You have now seen how MERFISH works. Now I want to take you on a tour of what all of that effort leads to the final results. We'll go from a fully reconstructed honeybee brain to cell clusters to subcellular patterns, and finally to some of the biological insights we've uncovered. By the end of this video, you'll be able to recognize the main outputs of a MERFISH experiment in the honeybee brain, understand how UMAP cell clusters connect to anatomical brain regions, and see how spatial patterns and molecular identities reveal new biology. Let's start with the 3D brain reconstruction. Here you're looking at the entire honeybee brain, rebuilt from 16 coronal sections, each spaced about 40 microns apart. Every single dot is a cell detected by MERFISH, and its color represents its molecularly defined cluster. With this 3D view, we can spin the brain, zoom in on specific regions, and trace structures like the mushroom bodies, optic lobes, and antenna lobes, all in one unified dataset. Now let's move to the UMAP projection. Each point here is a single cell placed according to its gene expression profile. Cells that cluster together share similar gene expression patterns, and we can color them by cluster identity. Here's the dataset after aligning across sections. When we take those clusters and plot them back into their original coordinates, we get a spatial map. This shows us exactly how different cell types are arranged within the brain and where transitions happen between molecular identities. One of the strengths of MERFISH is its subcellular resolution. We can see whether RNAs are concentrated in the soma, the cell body, or out in the neurites. For example, dopamine receptor RNAs like DOPR2 and DOP3 are often found in somas, while dopaminergic neurites contain RNAs for VMAT and TIHAD. This tells us not just what genes a neuron expresses, but also where in the cell that molecular machinery is located, giving us clues about where neurotransmitter synthesis and reception occur in a circuit. Zooming in on the mushroom bodies, we can break them down even further. By subclustering, we resolve distinct Kenyan cell subtypes, each with their own gene expression signature. These subtypes have been described before, but MERFISH lets us see exactly how they're arranged in 3 dimensions. Looking ahead, we're working on building a full 3D aminergic neuron atlas, a detailed map of neurons that use neurotransmitters like dopamine, serotonin, and octopamine across the entire honeybee brain. With this atlas, we'll be able to compare the brains of forager and soldier bees in unprecedented detail, looking at both their spatial organization and their molecular identities. Our goal is to understand how dynamic changes in RNA expression within these aminergic circuits contribute to the regulation of social behaviors, from foraging patterns to defensive responses within the honeybee colony.